Hello students, a warm welcome to all of you in ITLS Academy. In today's lecture, we will discuss about the bioinformatics tool, different web server and different application used in a bioinformatics. Okay, so we start today's lecture. First, we understand about the NCBI. Okay, what is NCBI? NCBI is a national Center for Biotechnology Information. Okay, it is established in 4th of November 1999. Okay, this NCBI is a part of NLM and NIH. NLM, National Library of Medicine, sorry, National Library, NLM. This NCBI is also a part. NCBI is a part of NLM and NIH. NLM is a national NLM and NIH, National Library of Medicine, and NLM is a National Institute of Health. Okay, okay. This is NCBI. NCBI, what is the NCBI? NCBI is a National Center for Bioinformatics Information. It is established in 4th November 1999. It is also a part of NLM and NIH. NLM stands for National Library of Medicine, NIH stands for National Institute of Health. Okay, here we click on this first page. This is the home page of NCBI. Okay, here NIH, that is a National Institute of Health, then NLM, that is a National Library of Medicine, then NCBI, National Center for Biotechnology Information. Here is a login. We also log in on this page by the help of user ID password and login with Google. Okay, then this is the search bar. In the search bar, we search our desired database. Here, all databases are present. This is the assembly, biocollection, bioproject, biosample, books. All databases are present in this list. If we want to retrieve nucleotide database, then we click on this nucleotide and search our desired component in this bar. Okay, here this is a home page of NCBI. This is a resource list. These are the all resources which are present in the site of NCBI. Next, this is the all resources and information about these resources. First is the assembly. What is the role of assembly? If we does not know what is assembly, then we also read this information, and if we does not any word, any abbreviation, if any database, then here all details are available. We also read the information from this. Okay, we also download the information about these. If we want to know about the blast, then we click on this blast. Wait a minute, and this is the all about the information about the blast. And we also download this information, and we also download the blast software. And next, tools. These are the tools which are available in the NCBI. Okay, these are the all information about the tools. This is how to, these are the information how to submit the data, how to visualize the structure, how to find the expression pattern. All the information are stored in this site or in this page. For example, if we want to know how we find the expression pattern, then we click on this site, and this is the complete information about how we find the expression pattern on NCBI. Okay, all NCBI is a very user friendly platform. In NCBI, we easily access the every information. These is the all program access. These are the information of all program. If we want to access the information about the chemical and bioassay, then we click on this page, then we access the information about the chemical and bio assays. If we want to the information about the data and software, then we click on this page, on this link, and we found the information about the data and software. If we want to know the information about DNA and RNA, then I click on this page. If we want to know the information about the domain and structure, then we click here. Okay. If we want to know the information about variation, then we variation. If we want to know the information about the training and tutorial, then we click on this site. Okay, these are the okay. This is the source list of NCBI. Here we also submit our data. We also down transfer the data to your computer. Then find the document is related. Then research, analyze, develops. Okay, all these information are stored in this home page. Here is the popular resources also available. Okay, PubMed, Bookshelf, PubMed Center, BLAST, Nucleotide, Genome, SNP, Gene, Protein, and PubChem. These are the public, these are the very popular resources. Okay, then here is the NCBI news and blogs. Okay, this is the home page of NCBI. In this home page of NCBI, we easily access all information related to our desired component. For example, if we want to know the information about the nucleotide sequence of P53, then then I select nucleotide from the all databases, then search the P53 and search. Okay, here the next page will open. Here this is the species list, this is the molecular type, source of data, sequence type. Filter out the we also customize the all resources according to our requirement. If we want to know the P53 gene only animal, then we select here only animal. If we want only P53 related to human samples, then we click on this human samples. Okay, these are the all p53 gene of human sex, human sapiens. Okay, if we want to know the molecular type mRNA, then I click on this mRNA. If we want to change the resource data, then we also easily change the resource data. Okay, if we want to clear the all filter, then we click on this clear all. Okay, now our all filter are clear out. Okay, here in the in this search box, we found the detail which are searched on this NCBI page. For example, first we search P53 gene in all field. Okay, then we search the Homo sapiens search gene. Then we search the P53 gene in Homo sapiens. Okay, here the or search related is mentioned. This is the history recent activity which are performed on this page. Okay, then we simply click this P53 Homo sapiens gene. Here the all databases is present. This is a GenBank format. If we want to change the format, then we easily click here and change the format. Okay, if we want this information in graphical form, then we click this graphical. If we want the information, this is the loading, this is the graphical representation of our result. Okay, if we want to access the information in faster format, then we simply click on this faster format. Okay, wait a minute. This is the faster format of Homo sapiens P53 gene. Here is the information of locus, definition, access number, version, keyword. All information is present in this GenBank format. Here, if we want to change some reason, we also change here. If we customize some views, then we also customized. If we want to perform blast, then here we click this blast button and perform the blast. Here we also find the information what is the blast doing. If we perform the blast, then we find out the reason of similarity between the sequence and other sequencing using the blast. Why we will perform blast? Here the information is available. We perform the blast to find out the similarity between the sequences. Okay, if we want to pick this primer, then we design and test the primer for the sequence using the primer blast. Okay, if we click on this pick primer, then we get this result. If we click on this highlight sequence feature, then we get this information. Open the highlight feature bar and highlight features annotation, and from the feature table of the recorded, this highlight feature bar can use to navigate to act to and highlight the other feature. Provide the link, display the highlighted region separately. Link of the feature databases will also highlight corresponding region of the sequence and more information. Okay, suppose here, this is the detail information, this is the highlighted region. If we want to search, if we find the subsequences or pattern in this sequence, then we click on this find region, and here we search our desired result and finally find. These all information are available in this page. If we want to download this, then simply click on to send it, then complete coding region या gene feature. This is completely depend upon our requirement. Then file format, then click the file format, GenBank Astra, which types of in which if we want to download the sequence, we also select the format according to our requirement. Okay, this is little bit about the NCBI. Okay, now we understand the next web server, that is a EMBL or data resources. Okay, for EMBL we search the EMBL EBI homepage. Okay, this is the EMBL or EBI homepage. The homepage of EBI. Here the all databases are present. If we want to know the information about the nucleotide sequence, then we click on this. Then we click on nucleotide sequence. Then we search our result. For example, P53. Then we search it. It will take some time. Here we also customize according to our requirement. In for related the P53, here 88,895 sequence and information or resources are available. Okay, we also select our we also customized these page according to our requirement. It will take some time. It will loading our result. Okay, these are the nucleotide sequence related to the Homo sapiens. Okay, now we click on this first information. This is the nucleotide or database. This is the nucleotide information from the. This is the name of organism. This is the accession number. This is the mole type. This is the topography. This is the base count. This is the data class. This is the text division, keywords, and MDF checksum. Okay. Here we also, this is a view detail. If we want to see our result in EMBL format, then we click on this. This is the EMBL format. If we want to access our information in FASTA format, then I click this is the FASTA format. If we want to download in EMBL, then I click on this. If we want to download in FASTA format, then I click on this. This is the cross reference. This is the publication source. This is the ORCID data claim, and this is the sequence version. Here view. Also access the all information related to our result. Okay. Here this is the in here we click the then we switch the home page. Here we submit this is a checklist. Then search. This is the rule escape. This is all about the NCBI. This is the support system. Okay. This is all about the EMBL. Now we discuss the next bioinformatic tool, that is a Emboss. Okay, first we search Emboss. Now we discuss the next tool, that is a Emboss. Okay, this is a software related to, or this is software is maintained by the created and maintained by the EBI, EMBL, EBI. The full form of Emboss is European Molecular Biology was open software suite. Okay, this tool is used to perform pairwise sequence alignment. Okay, what is the EMBS? EMBS is a pairwise sequence alignment tool. We perform the pairwise sequence alignment by the help of this Emboss. This Emboss tool is created and maintained by the EBI, EMBL or EBI. basically EMBL, EBI. Okay, the full form of emboss. This tool is created and maintained by the EBI. Okay, the full form of emboss is European Molecular Biology Open Software Suite. Okay, now in search bar we search the emboss. Okay, then we click this EMBL EBI emboss link, and this emboss needle home page will open. Okay, here we found a two section. In first section we paste our first sequence, then we paste here our next sequence. We also choose the file from our computer. Okay, then here are the some parameters. This is the output format. This is the output format, in which format we want to extract our result. We also change this format according to our requirement. Okay, here I will give, here we give us some job title for our pairwise, for our PSA or pairwise sequence alignment. Okay, here we give some name. We give the name such like our project or bioinformatics project, etc. Okay, this is the overview about the home page of emboss. Okay, how this is the overview about the emboss. Okay, how this emboss tool is run, we discuss this in our incoming video lecture in detail. Okay, next we discuss the cluster omega. Here we search cluster omega. Okay, this tool is also created and maintained by the EBI. Okay, this cluster omega tool is used to perform the multiple sequence alignment. For pairwise sequence alignment we use emboss, and for multiple sequence alignment we use this cluster omega. Okay, here this is the home page of cluster omega. Here we put our query sequences. We perform the multiple sequence alignment with protein sequence, with DNA sequence, with RNA sequence also. Here we also choose the file from our computer. Okay, here this is the output format. Here we also give some title from our MSc. Okay, these are the some other parameters which are present in this tool. We also change these tools according to our requirement. Okay, here this align input, distance metric, yes or no. These all parameters we change according to our requirement. Okay, this is overview about the cluster omega. How this will work, we will also discuss this in our incoming video lecture in detail. Okay, now we discuss the next tool that are used to find out the gene. Okay, basically GeneScan is the gene prediction tool. By the help of this GeneScan we predict the gene. Okay, GeneScan is used to identify the complete gene structure or genome. Okay, next, this is the home page of GenBank. This is maintained and created by the MIT. Okay, this is the home page of GeneScan. Okay, here we sequence the name. Here we also choose the signals or parameter according to our requirement. Okay, DNA sequences from our computer, and here we also paste the DNA sequences. Okay, then run the GeneScan, and finally we find out our result. Okay, one point is very important, and remember this point. This in Imbos, in Cluster Omega, in GenScan, in Blast. In to run all these tools, we use the faster format. Okay. Now we discuss the PDB. That is a PDB. What is a PDB? PDB is a protein databases. In protein, we found out the protein structures in 2D format, in 1D format, in 3D format. For example, this is the home page of PDB. That is a protein databases. This is the information about the structures which are present in under the PDB. This is the depository tool. This is the searching. This is the visualization, either analysis. This is the downloads. This is the ligands. About the documentation. This is the carrier. This is the COVID-19. This is all about the this, and this is the search bar of PDB. For example, if you want to if we want to search the 3D structure of insulin, then we search here. For example, here I search P53 gene, P53 protein. Okay. Then search, loading. These are the different. This is the query sequence in 5123 structure of PDBs is input in this PDB software. Here we also change and customize the information according to our requirement. Here, if we want to search the P53 protein related to human sequence, then I select this. Okay. If we if we want to predict the P53 gene related to the E. coli, then I click on this. Okay. This is the taxonomical respect. This is the taxonomy. We also choose the taxonomy according to our requirement. This is the experimental method. If we want to extract the 3D structure from X-ray crystallography, then I select this. If we want to do the 3D structure that is visualized by the electron microscopy, then I select this, NMR, then this. Okay, these all parameters are changed according to our requirement. Okay, now this is the first. This is a Geographish P53 gene. Okay. Now I click on this. This is the PDB 3D structure of P53 gene. This is the tetramerization domain of Geographish. This is the PDBI ID. This is the classification. This is the organisms expression system. This is the all information available related to this protein. We also download this file. We also display this file. Okay. And we also here we also download this file in different format. Okay. In what? Okay. And this is the structure summary. This is the structure. Here is the annotation. This is the experimental values. This is the sequence. This is the genome, and this is the version. Okay. These all information are present in this PDB. Okay. Now we discuss the next tool, that is a Swiss model. Basically, we use this Swiss model to perform the homology modeling. Okay, this is the home page of Swiss model. We, if we want to perform modeling, then we click on this start button, and either we paste the protein sequence and start the modeling. How this modeling is performed, we discuss this in our incoming lecture in detail. Okay, now we discuss the next tool, basically next software, that is a mega 11. Basically, we use this mega 1. Mega 11 is used to perform the basically to construct the phylogenetic tree. Why will we construct that phylogenetic tree? Because we want to know the information about the evolutionary relationship between the species. That's why we construct the phylogenetic tree. For the construction of phylogenetic tree, we use this software. Okay. How will we use this software? We also detail this in our incoming video lecture. Here I will tell you how to install this software in your PC. Okay. First, you open this software. This is the home page. This is the MEGA. Then full form of MEGA is Molecular Evolutionary Genetic Analysis. This is the tutorial related to this tool. This is the feature of this tool. This is the documentation. This is here. We also submit the feedback. Okay. Here we want to download the Mega tool for Windows. That's why I select this Windows. If you have Mac, then you also choose MacBook. Okay. But here I install this app for the Windows. Then graphical command line also graphical, but I choose this graphical. Then bit 62-bit and 32-bit. Another version, older version also, but here I choose 64 version. Now we click on this download button. Okay, this is the information about this Mega tool. This is the grant permission. This is the installation instruction, modification, etc. And then you accept. Click on this accept. Then here you also fill some information. You are instructor, students, or professor, and others also. Here, what kind of institute is. Here you put the institute name, then college and research institute also. Just say for example, here I belong to India. I am a researcher. I work on ITLS Academy. Here, I students. Other I also put there ITLS. Okay, then I download this tool. Okay, now this tool is this setup is installed in my PC. Now simply I set up this tool in my PC, and this procedure is very simple. But in my PC, this tool is already installed. That's why I cancel this. Okay. And how we use this tool, we also discuss this in our incoming video lecture. Now we discuss the next tool, that is a Swiss viewer. For the visualization of protein structure and for the construction of Ramachandran plot, we use this Swiss viewer. Swiss viewer basically is a type of software. To run this soft to run this so to perform to use this software, first we download the Swiss viewer. For the sound downloading the Swiss viewer, we click on this site and simply click on downloads and simply download this. Okay, here downloading is start. Okay, and the downloading of this software, this tool is also already downloaded in my PC. That's why I cancel it, and I will show you the software related to. This is the setup of Swiss viewer. These all are the files that are downloaded. Then you click simply the downloaded button and download the file. Okay, and we easily run this file. Okay, and then simple click on download and download this software in our PC. Okay, and how this Swiss PDB viewer is run, we also discuss in detail in our incoming video lecture. These are the tools, these are the software which are required in our incoming training to perform the experiments or practical related to the bioinformatics. Okay, first first we required how to use NCBI. We use this NCBI to extract the protein or to retrieve the protein and nucleotide databases. We use PDB for 3D structure detection. Next, we use Imbos for pairwise sequence alignment. We use Cluster Omega for multiple sequence alignment. We use Swiss model for perform homology modeling. We use Swiss viewer for the visualization of protein structure. Now we discuss the Chow-Fasman for the secondary structure prediction of protein. We also use the Gore method. Okay, now we discuss about the Gore Chow-Fasman. Okay, now we search the Chow-Fasman. This is the home page of Chow-Fasman. Here we simply submit our protein sequence in FASTA format and we also get our result. These are the home page, this is the blog, this is the tools, this is academic, contact and mail detail. Okay, next Gore tool. This is also a Gore tool for the protein structure detection. Basically we extract the secondary protein structure detection by the help of this Gore. Okay, these are the tools which are used in our training to perform the bioinformatic experiments. First, for the extraction of nucleotide and protein sequence, we use the NCBI. For the visualization, we use the SysViewer. For prediction of 3D structure, we use the PDB. For performing the pairwise sequence alignment, we use Imbos. For MSA, we use Cluster Omega. For Swiss model, first we for Swiss model we use the homology modeling. Swiss beer for visualization, Chow-Fasman law for 3D for the secondary structure prediction. And for construction of phylogenetic tree, we use the Megha 11. Okay, for we use first we download these software in our system. First software this and second software is this. Okay, how we download this software, I will provide the in description. I also provide the link related to these bioinformatic tools and software that are used in our incoming bioinformatic training. Okay, if you have any doubt then comment on comment section. Okay, please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share and subscribe ITLS Academy. Thank you.